So I'm at ASH 2018, and it's exciting to present CAD-01, or the THINK trial, which is a CAR-T trial focused on patients with hematologic malignancy, particularly patients with acute myeloid leukemia. So CAD-01 actually takes the activating or predominant killing receptor in natural killer cells known as NKG2D, and is then linked to CD3 zeta. And then within the T cell actually uses endogenous DAP10 as the co-stimulatory molecule for activation. NKG2D ligands can be expressed actually on a wide variety of cancers and are really absent to minimally expressed on normal tissues. And we've actually seen in AML 100% of patients having at least one NKG2D ligand, which there are a total of 8, MIC-A, MIC-B, and the ULBP family. The design of the trial is a traditional phase 1 dose escalation. We looked at 3 different dose levels, starting at 3.0 times 10 to the 8 cells per injection up to 3.0 times 10 to the 9 cells per infusion. Patients get 3 infusions spread by 2-week intervals, and then get disease assessment, and then have the option of getting another infusion. This trial is a little bit different in that we do not have any preconditioning chemotherapy. So basically patients get apheresis and 3 weeks later get their initial infusion. As far as safety, which is the primary endpoint in the phase 1 portion of the trial, overall is safe and well tolerated. We do see cytokine release syndrome actually occurred in approximately 42% of patients, with only 3 out of the 14 patients, or 21%, having grade 3 or 4. All patients had manageable CRS with tocilizumab leading to rapid resolution of cytokine release syndrome. The time or the onset to CRS is a little bit different in our patients. So the mean onset was 1 day, and typically short-lived, often less than 6 days. We really have not seen any off-target organ toxicity, and again, other side effects kind of pretty traditional in the spectrum of cytokine release syndrome. As far as early efficacy, we had 8 patients that had completed all 3 infusions and had disease assessment. We did have 3 objective responses, 1 CRH, and that patient was actually bridged to allogeneic transplant, and that response has been now durable for 18 months post-transplant. We additionally have 2 patients that achieved CRI. Additionally, out of all 8 patients, 5 out of 8 had some anti-leukemic activity with a significant blast reduction. So in conclusion, our overall NKG2D CAR or CAD-01 has been safe and well tolerated, again with 5 of 8 patients having some degree of anti-leukemic activity, including 1 more exceptional responder. Again, that patient's alive 18 months. I think our goal is really to optimize the activity of CAD-01, and that's through several different mechanisms. One is looking at decreasing the interval between infusions to 1 week, so that is going to be in the current amendment of the THINK trial. And then we have 2 other trials, one that is now open and one opening, one called EpiThink, looking at standard flu-cy preconditioning. Hopefully that may further augment the efficacy. And another is looking at combination with azacitidine.